Welcome to the project booth for Team A7160, a multi-scale open knowledge network for precision medicine, at the 2021 NSF Convergence Accelerator Expo. We would like to share with you our project team's work utilizing Spoke, the scalable precision medicine open knowledge engine. Spoke today brings together 37 specialized databases into a biomedical knowledge network with 3 million nodes and 15 million connecting relationships. These relationships allow us to span individual biomedical domains whose data are currently siloed across thousands of public repositories. In addition to expanding the network, the team is developing tools that are aimed at serving specific user groups, including citizen scientists, who want to better understand how these different aspects of biology can affect them and their loved ones. Biomedical researchers, who seek to generate hypotheses that can be experimentally validated. Clinicians, who want tools to aid in diagnosing and prescribing treatments that are specifically matched to their patients. And investigators in the pharmaceutical and biotechnology sectors, who are seeking to develop new therapeutics and potentially repurpose existing ones. Let's hear from the team at Indiana University, who are working to develop a network visualization tool for the general population.
Today I'm presenting the Spoke Visualizer developed at the Cyber Infrastructure for Network Science Center at Indiana University. Our visualization uses data available from Spoke. There are 3 million nodes and 30 million edges. And you see an overview network here that shows the different node types and edges that connect the node types. We're primarily going to focus on foods and diseases in this visualization. So we created 4 different trees of from the data connecting Alzheimer's, coronary artery disease, and epilepsy to foods that may interact with that disease. And then we weight those nodes according to betweenness centrality so that we can see the more important nodes first as we zoom in through this zooming interface that I'll be demonstrating. So we first come to the Spoke Visualizer, and we can be presented with the overview network. Nodes are size coded by the number of nodes in the knowledge graph of that type, and edges are size coded according to the number of edges between these different node types. And so we first start off by selecting a disease we care about, say coronary artery disease, and then we'll type in a food that we maybe like potato, and search. So then the overview network updates so that you can see the path that you have to take from that disease, coronary artery disease, to the food, potato. So it goes through compound in this case. So now we can then get a detailed view by clicking this. And now we see a visualization of coronary artery disease and then all the foods that are connected to it and the path that it has to take to get to that food in the knowledge graph. And highlighted, you see coronary artery disease and potato. So as we zoom in, we get more and more relevant labels. You see folic acid between the two. And as I zoom in more, you'll notice that it brings in more edges and one more level, and you'll see that it connects to potato. So potato connects to folic acid, which connects to coronary artery disease. So then you can explore this user interface and find different interesting pieces. This is designed to be explored like a map. And so that's the Spoke Visualizer. Thank you.
Now let's hear from the project's principal investigator at UCSF about the Neighborhood Explorer, a prototype interface to Spoke, aimed at serving biomedical researchers.
Welcome to this demo of the Spoke Neighborhood Explorer. And in this demo, I'm going to show you what the interface can offer in the graphical user interface capabilities. So when you are in the main interface, there's a number of sample queries which you can show or hide that are ready to go. All you need to do is to click on one of them, and it will run a particular query. In this case, is running a query for the thyrotropin receptor protein, which you can obtain more information when you hover the mouse on top of any node or any edge. In this case, the nodes are represented as green circles, and the proteins represent the green circles, and drugs that affect or that bind to these proteins are represented as purple circles, and you can see what type of node this is. In this case, it's a compound. It tells you the properties, which database this comes from, and so on. So in the case of this particular example, you know that thyrotropin alpha, the compound binds the thyrotropin receptor, and the thyrotropin alpha in turn is being used to treat different types of diseases. In this case, several types of cancer, and you see which phase the drug is in, which phase of development the drug is, and which is the source of this particular edge. In this case, there's another link here to another compound, which in addition to binding to the thyrotropin receptor, it binds two other proteins, and here you see it represented in this way. So you can also hide or show different types of nodes, and this is handy when the network gets more crowded. And you can also zoom in and out by swiping your two fingers up or down the mouse pad. And you can also rearrange nodes in any particular way that makes visualization easier. Then if there's a node that you wish to delete, you can highlight it and then click delete. You can also redo the layout and zoom in. And then there's a number of options that are available to you when you click the options checkbox. And here it will allow you to bring additional node types or edge types as you determine. So for example, if I clear all and I want to say bring only protein nodes, and the type of relationship I want you to bring only gene encodes proteins. So then, if I want to know what the gene is, I leave this edge, and then I want to bring the genes encoded by any particular protein that I highlight. In this case, I only have two. I can highlight this protein and say extend. This will bring the gene encoded by this protein. If I highlight this protein and click extend, this will bring the gene encoded by this protein, and so on. I can continue extending the network with any node that I wish and any kind, and bring any kind of nodes or any kind of edges that I wish. And this could become, in a hypothesis-driven manner, very, very useful.
Let's take a closer look at a more specific biomedical researcher's use case.
We illustrate here the use of our biomedical open knowledge network, Spoke. Let's start with a generic case. A biomedical researcher is interested in nutrients that may modulate a monogenic metabolic disease, D1, which is caused by an overactive gene. And there are all these databases that one can consult. And the first question would be, what is a gene X that causes disease D1? You look that in a gene disease database, and you might get as a return, gene G1 causes disease D1. Now we want to know whether there are chemicals that can inhibit gene G1, which is overactive. So you go to a chemical database and you ask what chemical X affects gene G1, which causes disease D1, and and you might get as a return chemical C1 suppresses gene G1. Then you could use chemical C1 and develop a drug, or you can, for example, hope that there exists a food item that contains already chemical C1. So you go into a food database and ask, what food X contains a chemical C1? And you get as a return food F1 contains chemical C1. So there are all these databases that one can query separately in order to answer that big question of finding a way to modulate gene G1, which causes disease D1. But wouldn't it be nice if everything were integrated into a giant network so that you can, with one single query, find the interaction of disease D1 with food F1 and connect the dots? And that is exactly what we have done with Spoke, which has incorporated many biomedical databases to a huge network of millions of data points that are appropriately connected. So let's show this use on the case of Kawasaki disease, which is a complication in children with COVID-19, as you might have heard of. Kawasaki disease has long been connected with a protein in the blood called S100A12, that's from the literature. But we also observe now that patients with severe COVID-19 also have a very high level of S100A12, and that raises the question how all these three things, the protein, the disease, Kawasaki disease, and COVID-19 are interrelated. And so this is a typical scenario where we could use Spoke knowledge network. So is shown here in this network. So we have the protein S100A12, which is a node in Spoke. We have the Kawasaki disease, another node in Spoke, here in red, and we see the connection between these two, the protein and the disease, which is known from the literature. And here you see the molecular interactions. But we also have proteins from SARS-CoV-2 virus, and you see the interactions between these proteins with the protein S100A12. So we have now closed the loop, and we can connect and explain this empirical observation that you see on the right between COVID-19, S100A12, and Kawasaki disease with a molecular mechanistic pathway. So the basic idea of a biomedical open knowledge network is shown here. The goal is to connect the dots across domains. The dots are entities in different domains, and we want to have a seamless integration of all knowledge so that there's no need to know which database to consult, and we like ideally to discover new connections that have not been seen before. For example, the blue dots here are from drug research. Every dot here is a chemical. The yellow dots here are for molecular biology. Every dot here is a gene or a protein. And then we have also from clinical medicine all the disease and symptoms. For example, if SARS-CoV-2 virus or its S protein is shown here, it will be connected through Spoke to all other entities from different domains, and we can extract interesting pathways and further analyze them, for example, to explain observations or to develop new drugs.
How about we take a look at how the Neighborhood Explorer might help a researcher solve a specific use case?
Hello, welcome to this demonstration of the Spoke Neighborhood Explorer. In this example, I'm going to show you the neighborhood of the disease phenylketonuria. And we start here by entering the disease ontology identifier, which I pre-populated here. And when I hit submit, you'll see the immediate neighborhood of the disease, which is in the center. On green, we have the nodes that correspond to tissues or cell types that are affected by this disorder. And move them to the left. And then to the right, I have manifestations or symptoms of the disease. And when you hover the mouse on top of any node or any edge, it will tell you the provenance and some more properties from those. Here are drugs that are used to treat the disease. You can see here's a compound treats disease node, and here's the gene. So this is the gene which is defective in phenylketonuria. So now I'm going to explore a little bit more. I want to know which is the protein that encodes this gene. So I clear all this and only highlight protein, and I only highlight gene encodes protein edge. And then when I click expand or extend, I bring up the protein. Now, if I have the protein, I can extend this further because I know that it is an enzyme which is defective. So I can bring what is enzymatic, the catalytic activity of this enzyme. And this here is a monoxygenase catalytic activity. And I can go even further to bring the specific reaction that catalyzes. So you see catalysis reaction. When I extend this, it will bring all the reactions that are catalyzed by this enzymatic activity. Some of them are redundant, so I'm going to delete the ones that are redundant and just keep one of them. And then I can go all the way to identifying the compounds that are either produced or consumed by this reaction. And in this case, I'm going to eliminate this option. When I click extend, I'm bringing the compounds that are participating in that reaction. So here you see I can reconstruct perfectly that this reaction processes tetrahydropteridine plus oxygen produces tyrosine and then tetrahydropteridine. And then here you have a complete representation of all that is known about phenylketonuria in Spoke at the moment.
Let's hear now how the team envisions Spoke being used in a clinical setting.
Hi, my name is Kate Rankin, and I'm a professor in the Department of Neurology at UCSF, and I'm a member of the Spoke team. One of the important applications we're developing for Spoke is to bring the biomedical knowledge network directly into clinical care. I'm part of a team of investigators, including Riley Bove, Stefan Saunders, and Erica Schleimer, who developed a platform called Bridge, which is currently live at UCSF. Bridge is activated when a clinician is looking at their patient's chart in the electronic health record, where a button click opens up our application directly out of the EHR into a new window. Bridge is built so that data from numerous sources can be combined to create informative visualizations that impact clinical care. We have a number of APIs that bring back data in real time, including an API from the EHR itself, of course, but also from the radiology system that houses medical images, from REDCap and Qualtrics questionnaire engines, and also from other research tools and databases that store additional clinical and normative reference data so that we can use that to contextualize the individual patient's results. We now have an API to interact directly with Spoke. What this means is that Bridge can now extract key data points from an individual's EHR chart, such as results of lab tests, their symptoms, their medications, and then it can recode that data into Spoke entry points and derive a patient-specific biometric signature. This allows the clinician to ask Spoke questions about the clinical and biological features of their patient in a way that has never before been possible at the clinic. Our current Bridge Spoke integration is designed for a relatively simple initial integration, specifically to determine how similar or different the individual patient's biological profile is from that of other patients with a particular diagnosis. The goal of this is to provide additional diagnostic information to the clinician that they can choose to integrate into their thought process about the patient as they're doing a diagnosis. However, you can imagine the many additional applications we can implement from this Bridge-enabled Spoke system. For instance, we can use Spoke to derive biological risk scores for individual patients so that we can better identify when their medical profile suggests that they're likely to develop a particular disorder in 1 year or 5 years. This can also be augmented if we give the clinician more information about why Spoke has suggested that the patient has this elevated likelihood of a particular diagnosis, including telling them which features of the patient's medical record are driving that suggestion. And this Spoke bridge integration has the potential to be used to guide treatment selection as well. You can imagine a scenario where a clinician has multiple effective medications to choose from. They're all appropriate for the patient's condition, but there are biological features of the specific patient that influence which of those medications is going to be the most effective for that individual. Spoke's deep biological readout of that patient can give the clinician the information they need to make the right treatment choice. So we're actively analyzing Spoke data to identify multiple applications of this bridge Spoke integration in different body systems and clinical specialties. And we're excited about the possibilities that this holds for revolutionizing information technology in the clinic. Thank you.
Finally, let's hear from the CEO of Mate Bioservices, the company launched to support commercialization of Spoke-powered apps.
Hello, welcome to Mate Bioservices presentation. In the last decades, an unprecedented volume of biomedical data has been generated. In addition, electronic health records have been digitized and made available to researchers. However, even if this information is today available, the application of machine learning and AI tools is challenging due to several reasons. The first one is the electronic health records have been digitized as paper. Therefore, it's difficult to obtain the data from those records. The second is that databases are largely unstructured and not easy to process by a computer. The third is that researchers have had several issues to interconnect these different biomedical databases and therefore obtain results. And the last one is that even in some cases where machine learning and AI has been utilized, you need to provide boundaries of biology of the human body to make sense of the result. Therefore, Mate Bioservices found a solution, and the solution is called Spoke. Spoke is a database of databases created by UCSF that encompasses 25 health-related databases, includes genomic, toxicologic, metabolic, and pharmacological data. Spoke offers a superior approach to machine learning and AI by allowing to embed data anonymously of big data sources, by using biology to constrain the results, by being scalable to add new databases that is generated later in later date, and by using real patient data that can be embedded anonymously and allows to predict real outcomes. We are a company that was born in the year 2020 to commercially harvest the power of Spoke and provide services to pharma and biotech companies. Mate Bio has obtained an exclusive license from UCSF for all commercial applications of Spoke. We have a very experienced team led by Sergio Arancini, the Distinguished Professor of Neurology at UCSF, at our R&D head, and myself with 25 years in the biotech and pharma industry. We provide two services. The first one is called Brick, and basically is a service as a service tool by subscription where you can actually navigate Spoke in a graphic context and enables you to evaluate indications, mechanism of actions, and side effects of molecules and diseases with a potential therapeutic use. This subscription service will be offered to different academic as well as pharma and biotech companies. The other service we offer is called Phoenix. It's a service for pharma and biotech companies to repurpose an existing drug or bring a failed therapy back to life. We use a proprietary technology that combines Spoke, a large patient database that includes 1.5 million patient data, and some specific machine learning capabilities that enable us to identify new indications for existing products in the market, or use clinical data of a failed product and try to find a cohort of patients that would bring the product back to market with a narrower indication. Thank you for hearing this presentation. Our goal is to help our customers unleash the potential of all healthcare data for the development of their new therapies. Thank you for hearing the presentation of Made Bioservices. Goodbye.
We hope you've enjoyed hearing about our project. Now we invite you to meet the team, and please feel free to ask us questions.